speaker.
Okay. So our next speaker today, I'm going to pull up the title of the talk to make sure I get this correct. But we have Dr. Christina Meritz, who is a neuroscientist and assistant professor of neurology at Kennedy Krieger and Johns Hopkins University. Dr. Nemeth Mertz's work is on pediatric leukodystrophies, including LBSL, and she began that work in 2016. Today, her work focuses on the use of mouse and iPSC-derived patient cell models to understand disease mechanisms and test therapeutics and therapeutic strategies.
Thank you. Can you see my slide all right? I hope you're seeing the right version.
That's good.
Perfect. Thank you. Okay, thank you. Good afternoon, good evening, everybody. Very happy to be here to share some of our recent data on AAV9 and ASO therapy for LBSL. LBSL has been talked about a couple times today, so I'm going to skip this slide completely. I think we're up to date, and Dr. Abdinoor just had a really nice overview of a lot of LBSL clinical symptoms and presentation. So what I just want to start here, this talk is focused on gene targeting therapies. And this is a point that was raised a little bit earlier is, you know, we understand the heterogeneity of LBSL and of course the different mutations and the compound heterozygous nature of this disease. So really having a targeted therapy for each patient proves a little bit difficult as there is so much variation. And we do know that there are many different mutations that are associated with LBSL. Dr. Abdinoor had this exact same image and pointed out this hotspot here in the second intron of the gene DARS2. And as you can see by the number in parentheses here, the majority of patients, and I think it's somewhere around 90% of patients, have a compound heterozygote presentation with one mutation in this intron 2 site. So this is a weak splice site in general in the normal population. And for LBSL, it is a particularly bad area for mutations. And this paper was published nearly 10 years ago now. So the number of mutations in general, I'm sure, has increased, but we continue to see this sort of hotspot. So this is going to be important for the rest of the talk. I want to talk a little bit about our model. I presented in the past on this, and today we're going to take it a little bit further, but we use a induced pluripotent stem cell model. These are iPSCs that are derived from peripheral blood mononucleocytes that are collected from patients here at the Kennedy Krieger Institute. We sent those PBMCs to Cedars-Sinai in Los Angeles, and they reprogrammed them into iPSCs for us. And then together with the help of Dr. Mingyao Ying here at the Kennedy Krieger Institute, who has developed a rapid lentivirus-based protocol to turn iPSCs into neurons. And so he does this a lot with dopaminergic neurons and motor neurons. And for the purposes of LBSL, we have differentiated our iPSCs into motor neurons. We did our best for a long time to do age and sex match controls, and we've recently come up with isogenic controls for many of our lines. And the data that I'm going to show today are sort of a mix between the controls and the isogenic controls. This is data that I've shared in the past, and I shared it last year too. So this is, I want to show you a general phenotype of our iPSC-derived motor neurons. This is the INCU site, similar to what Dr. Tenisma showed earlier. So this is just showing growth and neurite development arborization of these neurons. And so we can see with our LBSL shown in orange compared to the blue that these LBSL cells over time have shorter neurite outgrowth and then have fewer branches out of these neurons. And so this is something that we've shown we've observed a couple different times. And then we are continuing to do this for all of our different cell lines. If we grow these neurons in a microelectrode array plate and stick them into what we have is the Axion Biosystems MEA system, we can measure the spontaneous firing of these neurons over time. So what we generally do is we take this plate full of cells and we stick it into the machine for a couple hours every day and look at their spontaneous firing. And what we see over time and over differentiation of these cells is that the LBSL cells continue to have a reduced mean firing rate compared to the controls. And this has also been consistent. In terms of mitochondrial activity, this is seahorse data, which we're all familiar with now too. This is just showing sort of the baseline understanding of how to read the seahorse plate or the seahorse output. And what we're interested here is the baseline and of course the spare capacity. So the difference between the max respiration and the baseline is really important for these LBSL patients. And what we see across several different patient neurons is sort of a blunted response to all of these different stressors in the assay. Some patients are better than others in terms of having, or some cell lines are better than others in terms of having some of it a response for the max respiration. But when we quantify this in total, we see that the spare capacity is really reduced compared to the control cells, and in some cases, unfortunately, almost non-existent. If we look at the ATP production rate in these neurons, we see that glycolysis is pretty consistent. And of course, this is the differences that we see are with the mitochondrial produced ATP. So this is something that we're not really too surprised by. So this is sort of a general phenotyping of the cells, and this is what we're using to move forward with to test some therapies. Again, I'm showing this just because I want to point out, again, this weak spot right here. And what was touched on earlier is what happens with these mutations in intron 2 is that causes an exon skipping. So exon 3 is skipped, and that causes a premature stop site and no protein production. And what we do also know about DARS2, just by looking on Ensemble, and then also we have some transcriptome data to support this, that there are many different transcripts associated with DARS2, 13, and then roughly half of them produce protein. So these up here are all transcripts associated with protein, and then all these transcripts down here are associated with nonsense-mediated decay. And for the most part, what these nonsense-mediated decay transcripts have in common is that skipping of exon 3. So we know that these patients that have that intron 2 splice site and lack exon 3 have an overall reduction in DARS2 protein. And we've seen that in Western blots, and we've seen it in proteomics. But this is going to be sort of an important readout for our gene targeting therapies as well. And then one other point that I just want to make with this slide is not all of these transcripts are full length. So not all of them go from exon 1 to exon 17. Some of these end a little bit early. And so for all that we know, these proteins have some function and they're associated with fully normal DARS2, but there are a variation in these transcripts produced by everyone. So one more thing that we just want to look at as our readout for some of our gene targeting therapies is, again, how to quickly look at this intron 2 mutation site. So we very simply designed PCR primers that bind in a forward direction to exon 2 and in a reverse direction to exon 4. And this is going to produce two different transcripts. Either it's going to contain exon 3 or it's going to skip exon 3. And then if you put this onto a gel and just look at what you get, we see that the controls produce mostly a full-length product, and then they have a little bit of this truncated product. And like I said earlier, this is normal in everyone because that's a weak splice site. However, for the LBSL cells, this is an iPSCs right now, they have a reduced amount of that full-length product compared to controls, and then an increased amount of that truncated product, which we know is associated with reduced protein overall. So this general readout PCR is very simple, it's very easy, and this is going to be something that we use in our gene therapies. So really our interest right now is focused on improving the splicing of DARS2. That's sort of one of the things that we're sort of focused on. And I'm going to show you data for two different projects that we have ongoing in the lab right now, AAV9 therapy, so adeno-associated virus serotype 9, and then also antisense oligonucleotide therapy. And then so the data that we have are very preliminary. We don't have a lot right now, but we have some exciting data that we think is promising and are looking forward to sharing with you. So just in general, many of you are probably aware of this, but the AAV9 therapy, AAV9 is a virus that's engineered to deliver DNA to cells. We have a single-stranded DNA that, of course, incorporates with the host genome to produce the gene and eventually a protein of interest. So this AAV9 can get into the cell, it gets in the nucleus where it basically goes through the normal process to produce DNA, produce mRNA and then protein. So the benefits of AAV9 therapy, of course, it's relatively safe. The AAV9 cannot self-replicate, so what you deliver is what you deliver. There is limited genomic integration. AAV9 in particular, this serotype has a high affinity for the CNS, so this is of course what we're focusing on here, and it crosses the blood-brain barrier, which is important for any sort of therapy that is going to be used to treat any sort of CNS disorder. There are drawbacks, of course. There is some integration. There's always a risk for integration. And that is, there are currently 2 FDA-approved AAV therapies, one AAV9, one AAV2. And this is something that, clinical trials have to work out, is dosing, which is often difficult to determine. But of course, there is some risk always associated with this in terms of cancer. And then another factor is that we often don't know how long it will last. In theory, it could last a very long time, but we're just not sure on that sort of thing yet. So a lot of things to consider with AAV9 therapy. For the most part, I think for many conditions, the benefits outweigh the risks, but that is, of course, to be determined for each disorder and each patient probably. So in the lab, how do we study AAV9s? So we study them in our iPSCs and our iPSC-derived neurons. We were fortunate to work with some friends and colleagues at the University of Maryland School of Medicine who do a lot of work with AAV. And they designed this vector for us to include the DARS2 gene. They made that and gave it to us, and we have been testing it in our lab. This is Adam. He is a technician in the lab who has been doing all these studies. He is leaving us soon for MD PhD program, but in the meantime is doing a lot of validation on the AAV9 work. So I have a little bit of his preliminary data here. In short, he takes the iPSCs, he derives them into motor neurons. He waits until these cells are mature motor neurons because we want to see that if we want to be able to see an effect in mature cells. That is what would be happening in the patients. And so at day 16 after differentiation, we consider roughly day 12 to day 14 to be mature. On day 16, he gives the AAV9 in various doses, incubates them for 10 days with the cells, and then we do qPCR to look for that effect on gene transcription. And then he also imaged every day to look at neurite outgrowth. The data that I have, again, they're preliminary, but eventually the endpoints that we're going to be looking for will include Western blot, looking at protein. So we want to, of course, see if we can change DARS to protein. We'll look at that MEA activity to see if we can affect spontaneous firing. I have a little bit of mitochondrial seahorse data to show you. I have a little bit of the neurite outgrowth to show you, and we're going to, of course, do this several times to confirm that. And then also we're going to look at lactate as sort of an output for mitochondrial function and stress. So first, looking at the gene expression. So this is the qPCR data. And I'm going to just walk you through this graph. It's a little bit complicated. So for 2 different LBSL patients, we have untreated and AAV9 treated results in the neurons. And then everything here I've normalized to exon 17. So this would be showing a full length transcript. So exon 1 to exon 17. But we know that because not all transcripts are full length, not all have exon 17, there is a variation for the amount of exon 6, essentially, that transcripts that include exon 6. So for this particular cell line, there are more transcripts with exon 6, and then in this particular cell line, there are about the same number of transcripts that include exon 6. But if we look on exon 3, where that splice site variation is and where that exon is often skipped, we see that in both of these lines, there is a significantly reduced amount of exon 3. So that would suggest, of course, that the majority of these transcripts lack exon 3 and do not produce protein. So if we treat with a high dose of the AAV9, so this is a high dose just to sort of make sure that it's working, we see a significant increase in the transcripts that express all of these exons. And you can see this is a almost, too much of an increase here that we're seeing. So in both of these patient lines, we see increased exon 3, exon 6, and exon 17. So the next step here is to look at protein to see if these transcripts are actually going through to make protein. And it's also important to note that This particular cell line here does not contain that intron 2 mutation. They do have this reduced exon 3, which is interesting, and it does work to increase gene transcripts because you're replacing the entire gene. So, of course, this is sort of a blanket approach for any patient and any patient combination of mutations. If we look at the neurite outgrowth, so this is just looking at imaging. So this was 1 image per day. This is a fairly blunt approach for this as well, instead of the by hour. This is day 1, 5, and 9 that I've summarized here. The lightest pink is no treatment, and then the low dose is red and a high dose is in purple, and this is just looking at 1 cell line. And so what we see that the high dose here translated to the greatest change in neurite outgrowth. So this is just looking at area, and you can see if we zoom in on two of these days, day 9 no treatment versus day 9 high dose AAV, you can see there the degree of axons and the neurites in the high dose versus the no treatment. So this is definitely a significant change, and this is something that we're going to continue to test in multiple cell lines. And then finally, looking at some seahorse data, so looking at the mitochondrial function in the AAV9, this is a low dose, and we have 2 cell lines again. So the light pink and the light blue are 2 different patient lines. And again, you can see the baseline in the first sort of quadrant, and then FCCP marks for the max respiration. And with our low dose AAV9, the red is matching with the light pink, and the dark blue is matching with the light blue. You can see that both of these lines had increased in baseline oxygen consumption here, and then also increased max respiration in this third quadrant here. Again, these are preliminary, and we will continue to test this sort of with different conditions, optimizing the dosing and see how that works across different cell lines. And then finally, one thing that we're also going to be doing in our mouse model is testing the AAV9 as a proof of principle and also looking for toxicity in mice. We have mice that lack DARS2 in a subset of neurons in the cortex and hippocampus in the frontal lobe. And so these mice are presented on this, and this is a paper we published a few years ago. These mice experience a progressive loss of these neurons in the cortex and in the frontal lobe. And you can see that in many of these pictures here. And they have a significant phenotype where they have this rapid increase in activity that starts at around 22 weeks of age. And so this coincides with the degeneration of the brain, the cortical regions. And so what we've done is we have, in a handful of mice already, we've injected this AAV9 to the ICV, intercerebroventricular, so into the lateral ventricles. And we hope that that AAV9 has sort of spread to the areas of the affected neurons. And we're going to see if we can interfere with the sort of behavioral progression and perhaps change the trajectory of this degeneration. So right now, we're still too early in the timeline of this, so we have yet to see if we're going to see a divergence of the behaviors or not. But hopefully this is something that we can update you on next year or sooner. So back to the other method, so antisense oligonucleotide therapy. This is a figure from a review paper written by Manu Amanat. He is a postdoctoral fellow in the lab, and he has been doing the work on the ASO. So he recently put out this paper with some really great figures. So basically what I want to show here is that there's multiple mechanisms of ASO action. So there's multiple ways that these antisense oligonucleotides can work. These are short strands of RNA that can go in and through a variety of different mechanisms interfere with transcription to ultimately interfere with protein production. And so you can see here translational arrest, destabilization of the mRNA, open reading frames, and then what we're going to focus on here is splicing enhancement and splice silencing. So this is really the mechanism that we are manipulating in LBSL. So what happens here which is particularly related to LBSL because of that weak splice site, is we can sort of trick the machinery into enhancement signals, and there are also splice silencing signals. And so if we can enhance the enhancers and silence the splice silencers, we can sort of fix the way that the protein machinery reads that particular chunk of RNA. So this is not a new idea, ASO therapy for LBSL. This is a paper from Dr. Vanderknap's group back in 2012. And so they used a variety of different cell types and constructs in which they induced DARS2 mutations in a variety of cell types. And so what they're showing here is if they can treat these mutant cell lines, so mutant A and mutant B with different oligonucleotides, they can change the ratio of this readout. So this is similar to what we had before, where you're including exon 3 or having exon 3 excluded. So if it's a bigger band or a bigger size, then we're including exon 3. So if they add this oligonucleotide, they can actually improve the number of transcripts that include exon 3 and increase, hopefully, protein production. So what they did in this study was to produce an ASO that has a tail, and this tail spreads into exon 3, which actually enhances the splicing machinery to more accurately recognize the splice site to include exon 3. What we have done is we have worked a little bit more downstream, and we are silencing a splice silencer. And so we have designed many different ASOs to target this region, and basically you just have to screen them all to see which ones work the best. But if you inhibit that splice silencer, you can hopefully cover up this area and then produce the transcripts that are full length. And so this is a picture of Dr. Amanat, who did this work and had that review paper. And so this is just a brief overview of his methods. And he did this a number of different ways, both in iPSCs and in NPCs, and also, in the stem cells, in the neuroprogenitor cells, and also in mature neurons. And the data that I'm going to show you today are from NPCs and also from neurons. And so in these studies, the ASOs are added to the cell media. They're allowed to incubate for 48 hours. And then RNA is either immediately collected for qPCR, or in one of the data plots that I'm going to show you, we waited for 7 days after that 48-hour incubation and measured lactate. And similar to the AAV9 therapy, the endpoints here are very similar. Gene expression, of course, protein to confirm that this gene effect is having an effect. We'll look at MEA activity, we'll do seahorse, neurite outgrowth, and then confirm findings with the lactate. So this is showing 3 different ASOs. The ASO1 is the one that we designed that is inhibiting the splice silencer. ASO2, for the purposes of this study, we recreated the Vanderknap ASO with the tail to see if we could recreate and replicate their findings. And then we also did a cocktail approach. So Manu had read in the literature that oftentimes a combination of ASOs seem to have an additive effect. And so we figured it was worth a try to also look at that. What you're seeing here is data from 3 different patient lines. So we have one clump here, another patient here, and another patient here. And as you can see, this is control. So we're normalizing now to control amount of exon 3. So this is actually showing the inclusion. So this is the number of transcripts that include exon 3. That is reduced, as we expect, in all of our patients who receive no ASO. And then with either a low or a high dose of ASO, in either one, the high dose seems to increase the number of transcripts that include that 3rd exon. And for the final, for this 3rd patient, you can see it had more of an effect where all of these ASOs raised this line's exon 3 inclusion back to control levels. In any case, all of these increases are an increase from that patient, that line's baseline. So these are all sort of exciting data for us to continue to explore. And then finally, looking at lactate. So this is lactate that's measured in the media. This is 7 days after therapy. As you can see in the control, so again, we have our 3 different LBSL patients, and in the control versus the gray bars for each patient line, you can see that lactate is increased. And with the low dose or the high dose in the hatched bars, we can see a reduction of that lactate in the media. So again, this is all sort of data that's pointing in the right direction. This is very preliminary, very early, but we're hoping that this is something that we can continue to validate. In terms of delivery, I didn't talk too much about this, but these are all in vitro studies. So of course we can just give it to the cell media. In the mice, we did a direct ICV injection. But for both of these therapies, it's very important to deliver to the site of activity. In terms of LBSL, this would be most effective as an intrathecal. And as you can see here in parentheses, there are drugs, there are ASO therapies. This is relative to ASO studies. There are drugs that are FDA approved that are delivered by each of these routes. So this is not something that is a new concept for treating disease. And again, for LBSL, it seems that it would be most effective, given the spinal cord effects, that it would be delivered intrathecally. And then finally, I just want to sort of go back to this idea, and this is something that Dr. Goldstein touched on this morning too, is that all of these diseases are very, very different. And so while this approach seems feasible for a disease like DARS2 or a disease like LBSL and intrathecal delivery, many of these ARS disorders and mitochondrial ARS disorders especially affect many different organ systems. And so having that direct delivery system or targeted approach might be a little bit more complex. So I think the title of this article, when a common biological role does not imply common disease outcome, is very apt for this set of diseases. And it's always something that needs to be taken into consideration when discussing treatment strategies. Just to wrap things up, we have very preliminary but important and promising data using both the AAV9 and the ASO therapy on LBSL patient cells. Work is still ongoing to measure cell function after therapy. So again, looking to see how mitochondria are affected, how electrical activity and growth are affected, because that's really what's going to determine whether or not this is something that is worth pursuing. The mouse studies are underway for the proof of principle and toxicity studies of AAV9. We're really excited to see how these unfold. We also have 2 different mouse models right now that one of which in the past few days has started to show phenotypes. So we're really excited to be able to explore that and then hopefully test some of these therapies there as well. And then despite this focus on therapy, we always have multiple balls in multiple courts. And so we are still looking at trying to understand mechanism of LBSL and of DARS2 dysfunction. So we have studies that are ongoing, looking at sort of DARS2 interactions and how DARS2 might be dysregulated across these cells. So hopefully that's something that we'll be able to share with you in the future as well. So with that, I'll wrap up. I'm not sure I said this in the beginning, but I need to, that all of this work is made possible by CureLBSL Foundation. So we've worked with them for several years now. It's been a wonderful partnership, and we've made a lot of collaborations and a lot of headway to study LBSL. And of course, the lab, Dr. Fatimi, and as Adam and Manu in the lab right now, who've done the most of this work, and our patients and families always. So thank you, and I'll take any questions.
Thank you. That was a really exciting, amazing talk. We do have one question in the chat. Eric Williams is asking, is your mouse model using mutations of a knocked-in human gene or equivalent mutations in the mouse?
So that's a good question. So the mouse is actually a deletion. It's not a complete knockout. So any complete knockouts are not viable or not compatible with life. These mice have a deletion of exon 3, and so they don't produce DARS2 protein in a subset in CAMK2 neurons. So many people have tried to knock in human mutations and for whatever reason have had difficulty doing this. There are groups still trying. We wish them the best. I hope it works out because it would be wonderful for everybody. But that sort of thing has proven very difficult. And we've tried neural stem cells. We've tried oligodendrocytes, and we just don't see the same effect that we do with these particular mice. So.
Thank you. And Calvin Lee is asking, you observed that LBSL iPSC got an increased truncated band and decreased full-length gene compared with control. How often did you see such two-band ratio patterns of disease progression and treatment evolving effects in patients at transcript and protein levels? What is the functional differentiation of the two variants in spatiotemporal manners in vivo in different patients?
That's a lot. So that pattern of increased transcripts lacking exon 3, that's something that we see consistently. So that's something that's across the board, especially in the patients that have that exon 2 or intron 2 mutation, which is I think 5 out of the 7 lines that we have, maybe 4 out of the 7. So that's something that we see frequently. In terms of how that affects the spatial arrangement of the complexes, I've looked into this information. I've read many papers that study missense mutations of DARS2 and how that affects structure and how that affects all different aspects of DARS2 functioning. It doesn't seem like there is a across the board answer to that question. I think each of these mutations has some minor effect on structure, on binding, but it's my thought that those, that's not the major culprit here. So that's so I mean, for that reason, we haven't really pursued it any further. I would be interested to know, but I also think that there are other mechanisms that are sort of resulting in these disease phenotypes. So I'm not sure I answered all that, but yeah.
Oh, I think you hit the high points. One more from Eric Williams, though. Are you seeing the AAV leave the CNS and enter the general circulation?
So this is still really preliminary. It was only a couple weeks ago that we did those surgeries. That's something that we can certainly look at. We haven't the mice are still alive, so we haven't looked at any of that yet. The AAV9 does have a GFP associated with it, so in theory what we can do is we can section different organ tissues, which we plan to do, look for signal, see if we can find it. We can do qPCR of any of these organ tissues to see if we have an increase in DARS2. These are all things that we are certainly going to do, and questions that we hope we can answer sooner rather than later.
Thank you.
Can I just jump in there quick on this question? Because we know from the gene therapy trials that have been already done in patients, for example, that typically the whole idea of injecting things into the brain or into the spinal fluid is because it's immunoprivileged. And so usually that virus does not get into the periphery. And that's a good thing, because that is a way to avoid building antibodies against the virus, which is, a common theme in systemic delivery of AAV9. So it shouldn't really, but it will be virus dependent. So each virus will be a little different.
Thank you all so much.